the plan is for Wednesday, I'll reserve some time for any needed clarification on what you've been studying for the exam. So remember, for the exam on Friday, it'll be two different portions of it, the multiple choice, the essay, and same idea as before. Make sure your laptop is charged up and you have it behaving appropriately. I've had at least one student in my other class, his laptop wouldn't connect to or wouldn't let him use the lockdown browser. So make sure everything's going okay with your laptops. You know what kind of flower this is? What plant this is? It has an association with the holiday tomorrow, or the celebration. Yeah, this is pumpkin. Pumpkin. Beautiful pumpkin flowers. So kind of hard to believe that something so delicate looking, I mean, it's big, but it's pretty delicate, that morphs into a pumpkin. But it does. So we have at the base of the flower, the ovary. And so that ovary tissue, if everything goes okay, you're going to have the ovary, the eggs getting fertilized, and then that will kick into fruit formation, fruit and seed formation. And so that morphs into that. Kind of cool. Something, pretty tough, and it all starts with a pretty delicate looking flower. I looked up, you can eat pumpkin. It's not just for carving. The jack-o'-lanterns, you can eat them. Very nutritious. The seeds you can eat. That looks delicious to me. The flowers as well. So this is just the flowers. You can eat the pumpkin though too, of course. And then giant pumpkins, probably seen maybe a little bit of that. They have giant pumpkin contests every year. It's a big deal in some circles. Reminds me of that movie, Wallace and Gromit, Curse of the Were-Rabbit, right? I love that movie. I think we should do this. Maybe not Lake Superior, maybe an inland lake. But this is a pumpkin regatta. This is on the East Coast. They get into it big time. And so they have to carve their own pumpkins and get in and do like an obstacle course, and under a time limit. It's a race. So I don't see many PFDs anywhere. So maybe a couple. Can't tell. Looks like some do and some don't. Enter at your own risk on this contest, apparently. I can post these slides. This is a time lapse of a giant pumpkin growing over its season. It's kind of fun to watch when you just want to veg out. Veg out. Sorry. That's technically it's a fruit, so I guess it's not so funny. And then if you want to really geek out, you can do this link, and it's like a little podcast on the physics of giant pumpkins. And then they have last year it was Travis Ginger who won the giant pumpkin contest. Remarkably, he did all of that pumpkin growing in Anoka, Minnesota, which is not prime area for giant pumpkin growing. That's really quite amazing. Weighed almost as much as a Prius, 2,500, 2,560 pounds. And so that's what it looked like. So this was before it made the trip. That's at his house in Anoka. And then this is at the official, I think the official weighing, where they all come together and they weigh it and they declare who wins. I think that happens in California. So he had to get that pumpkin safely to California and not have it break. This is a blurb from the write-up from last year. I like the third bullet. Minnesota has a great mid-year, but our spring in our parts is really, really tough. So to do it in Minnesota, it just shouldn't happen. So far, it seems he's got enough magic ingredients to make those special seeds sprout. It's like winning the Tour de France on a big wheel. He said, you can only hope, but it worked. So it's not easy. And he won again this year. So actually, he's won 3 years in a row. So I didn't put in his 2021 win, but he won 2021, 2022, and 2023. And this one's even more, 2,749, world record. So pretty amazing, but it's really flat like that. Yeah. They have to inspect it and make sure it's all intact, and then they weigh it. So that's a lot of time and effort. So what did he won? I don't know if it says, oh, I didn't put in there how much. I think it was around 30 grand he won this year. And it was 23 grand last year. But I don't know if that, oh, like what, I'm assuming that's some amount of profit, but it'd be interesting to see how much it cost him to grow that thing. Time and money. Anyways, I thought you might be interested in that with Halloween coming around tomorrow. So, all right, I can post those if you're curious to read more of that. I'm going to stop share so it won't freak out on me, hopefully. Okay, so I'm gonna go back into the light reaction slide set, and we've been following that process. And so, let me reshare. Share. Okay, so we've been looking at the light reactions, and we worked through pretty much all the way through the Q cycle. So we looked at this idea of how light energy is striking both photosystem 2 and photosystem 1. We have the idea of that chlorophyll in the reaction center getting excited, boosting off its electron, and the first one's not being listed here. It's actually pheophytin that accepts it. And then pheophytin will give it to plastoquinones. And you can see it's the plastoquinones that once they get fully reduced, they will be able to, so fully reduced is the PQH2 form. So remember, plastoquinone has to have 2 electrons to be chemically happy. And then it also has to grab 2 protons from the stroma. And then it becomes PQH2 and it starts to mobilize through the membrane, thylakoid membrane, until it finds a cytochrome B6F. And then that's where we saw the Q cycle, where if you look at what's happening in the cytochrome B6F, you have the 1 electron going into the linear route, which we'll put on hold for a second. The other electron goes to cytochrome B, another cytochrome B, and then to a plastoquinone that's in this complex, the cytochrome B6F complex. And then that plastoquinone, so once it gets 1 electron, it's semi-reduced. So that's how they denote semi-reduced, but it needs 1 more. So everything has to happen again from photosystem 2 getting hit with light energy, that chlorophyll electron getting excited. All of that has to happen again to deliver another PQH2 into the Q cycle so that you have the second electron coming along. And once that semi-reduced PQ gets another electron, then it grabs 2 protons from the stroma. And so this Q cycle is, when you look at it and say, why would a plant, have evolved this mechanism as part of the light reactions? And really what it's doing is it's allowing for protons to kind of disappear from the stroma and get dumped into the lumen. And so they often refer to that as proton pumping across the membrane. And so if you look at that Q cycle, that's really what it's doing for the plant. It's allowing for this buildup of protons in the lumen compared to the stroma. And so it's a proton gradient. And so that's a good thing. We want a proton gradient, a nice proton gradient getting established during the light reactions. That's one of the big goals for the plant to accomplish. So that's setting up a situation that's going to be very good for the plant. And I'll talk a little bit more about that. Okay, so, but let's follow the linear path. So we said if there's the linear path, you have a plastoquinone will be putting an electron into the linear route, which is showing that it's giving it to an iron sulfur, which then passes it to cytochrome F, and then plastocyanin is sitting there. And so plastocyanin just kind of sits there and it's associated with cytochrome B6F until it gets its electron. Then it releases and it starts to move. So it's a mobile carrier and it will move. It's in the lumen portion of the thylakoid membrane, and it just kind of skirts along that portion of the membrane until it can find a photosystem 1. And then it has to like wait its turn. Once it gets to photosystem 1, it needs a situation where photosystem 1 has also been hit with some light energy, and the chlorophyll electron is excited in that photosystem 1. It gets boosted off to an acceptor. Now the plastocyanin can say, okay, here's an electron to replace the one you just lost, you just gave off. And so the plastocyanin will be able to hand off its electron only after photosystem 1 has been hit with light energy. Okay, so then as the electrons pass through several players in photosystem 1, what you have going on here in photosystem 1 is that you have that electron in the reaction center getting excited. So that's the asterisk idea. And then when it actually donates off the electron, then you write it as P700 plus. And now it's ready to say, okay, I need an electron. And plastocyanin says, yep, I got one, here you go. And then the chlorophyll in the photosystem 1 will be back to its normal state. So the one that it did donate off, though, due to light energy excitation, it goes through a series of donors and acceptors in the photosystem. And when you look at what's going on here, you have a series of donors and acceptors in the photosystem, and then right associated with photosystem 1 is ferredoxin. So it's sitting there going, okay, I'm waiting my turn here. I need an electron. It's in the stroma portion of the chloroplast. So once it gets the electron from the photosystem 1 players, then it can release off of photosystem 1 and basically go out into the stroma. So it's mobile, and it needs to find FNR. FNR is what's called ferredoxin NADP reductase. So they named it that because it associates with ferredoxin. It needs to grab an electron from ferredoxin. And then that NADP reductase, what they're trying to say there is that FNR will give the electron to NADP. So it's reducing NADP. And so when you have this process, after ferredoxin gives the electron to FNR, then FNR will find NADP plus and give an electron to that. And then we say it's been reduced to NADPH. So that is one of the huge goals of the light reactions, is to get that NADPH. And the reason for that is because NADPH is, they say it's strong, or another word is good, good reducing. So what that means is NADPH can now enter into other reactions in the stroma and say, hey, I'm really good at donating off electrons. And so let me participate in reactions that allow, I can do my part. I can donate off electrons into different reaction pathways to help those go to completion. And so we're going to see how NADPH ties into the next phase of photosynthesis, which is often called the Calvin cycle, which is another way to say it is carbon fixation. That just means you're taking CO2 and fixing it into a carbohydrate. And so NADPH is one of the main goals of the light reactions. Okay. I wanted to do that on the click. All right, so now we have NADPH. So basically we've completed the electron transport chain game from start to finish. And we end up with that NADPH. So now we have to go back to that proton gradient and think about that for a minute. So as this electron transport chain process is cooking along, this process is happening. happening. It's called linear photophosphorylation. So linear just means that if you look at the electron transport process, if you kind of the Q cycle, we don't really think about it this way, but if you just look at the flow of electrons from water to NADP plus, it's a linear route. And so what they're saying is as that electron is passing down the chain from donor to acceptor in that linear path, you get phosphorylation of ADP. So when they are talking about phosphorylation in that word, they're really referring to this idea of this reaction happening right here, where you have ADP and inorganic phosphate reacting. And then the photo part of that word just means it's because of light energy that all of this is happening. So they often phrase it this way: a portion of the electron transport chain energy is conserved as ATP. So when you, well, let's get it down in words first. So when you have this electron transport chain process happening, they say it's energetically downhill. Another way to say that is it's negative free energy. That's good. That means it can happen spontaneously. Negative free energy is good. And so this idea of as the electron transport chain process is cooking along, it's energetically downhill. What that means is it provides some free energy now to allow for that proton pumping across the membrane. So when you look at electron transport chain, it's almost always, I don't know if there's an exception to this rule, it's linked to this idea of proton pumping. Electron transport chain, proton pumping. Electron transport chain, proton pumping. You're doing it right now in your mitochondria. They're doing it in the chloroplast in this case. They also have mitochondria and they're doing it in that one too. And so we have this idea of if you can link that, create a situation with electron transport where it's energetically downhill, that allows you to have some free energy to get the protons pumped across the membrane, get your proton gradient. And that's actually what will then allow ATP synthesis to occur. So if we go back to, and then don't forget, when water is getting split or stripped of electrons, protons are getting generated that way as well. So that also contributes to the proton gradient. So let's go back to this big picture here. So what they're trying to show here is that as you have the electron transport chain process, you have this proton pumping across the membrane. You have protons building up from the splitting of water. The Q cycle too, the PQ that's involved in here also is grabbing protons. All right, so you get this gradient. I wish the artist had been a little bit more generous and put more protons on this side, because you get this buildup of protons in the lumen, less in the stroma. So there's your proton gradient. And when they say it's energetically downhill, if you go back to this image, maybe that would help you think about it. So this is like a so-called energy tower, so higher energy up top. So what they're trying to show here is that when you use light energy, you're really boosting the energy of those electrons. And then as they start getting passed down the electron transport chain, it's downhill, energetically downhill. So what that, if you see a drawing like this, it's like, oh, okay, so now there's some free energy in this process because it's going downhill energetically. That means there's a little bit of free energy that can be used for something. And it's almost always this idea of getting protons pumped across the membrane so you can have a gradient. I often think when I was learning this idea, I would often think about, trudging up the snow hill to go tobogganing, and that's kind of like, you have to put in some energy, light energy, to get up to the top of the hill, or you have to walk and drag your sled behind you. And then I had a pet dog that would, as I got onto the toboggan and started going downhill, so now I'm energetically going downhill and I go faster and faster, he would chase after the toboggan and jump onto it. I often think about him as the proton. That now the proton's coming along for a ride, he's going to be able to come across the membrane. So that's the analogy I used as I was trying to learn this process. And that was a long time ago I learned that, and I still remember that little story in my head. So it's just a really slick way to not only get your NADPH made, you get that all accomplished, but you're also linking it to a proton gradient, generating a proton gradient. And then once you do that, once you have those protons more in the lumen, less in the stroma, they have to go, they're going to want to diffuse across to even out the concentration by the laws of diffusion. So they're like, oh, I want to cross over again. And they have to go through the ATP synthase to get across. And as they do that, it actually causes these subunits that are hanging out in the stroma side, they actually rotate and generate a torque force that actually allows the ADP and the inorganic phosphate to react with each other. And then you get that lovely ATP. So it's quite amazing that they work this process and link it to this proton gradient. It's a very nice way to create this system that allows for ATP. And that's why, like, when I was first learning this, I'm like, wow, this light reaction stuff, it's kind of complicated. Why doesn't the plant just shorten it up? Seems like that would be more efficient. Just make it a shorter reaction pathway. But if you do, if you talk to the experts who've done the thermodynamics of this process, and if you theoretically try to shorten it down, they said it would basically cause the plant to combust, like just burst into flames, because it would be too much energy, boom, released all at once. And so this way, it's just like this little bits of energy captured through the electron transport chain process that allow for, a very careful reaction pathway. So nothing gets out of control, nothing bursts into flame, but you accomplish making NADPH and getting a proton gradient. So it's really quite remarkable. Okay, so we've worked our way from start to finish, including the Q cycle. Questions? Good for now? Okay, so that's as far as I'm going to take it for the light reactions, but now I wanted to kind of merge into the next topic, which should be the next slide set posted in the EDUCAT. And I wanted to look at how, okay, so now they've created this ATP and they have got their ATP, the NADPH. So, okay, now what? So now we're thinking about the carbon fixation reaction, which when they say that, they just mean they're taking carbon dioxide in through their stomata, and then they're going to make their carbohydrate off of it. And remember, it's not glucose. That's not the first thing they make. That's not the first sugar they make. It's so that's a little bit of a misconception. So when we look at the light reactions, it's important to notice where the products are getting made. They're in the stroma. You have NADPH and ATP in the stroma. And that's really convenient because now the carbon fixation reactions are happening right there in the stroma. So those molecules will immediately get to be fed in to the Calvin cycle. So this is linking the two parts of photosynthesis. We've already talked about the left part where the light reactions are cooking along. And then the dark part on the right side there, or the blue part, I should say, is the carbon fixation idea. And so they call it the Kelvin-Benson cycle. I tend to shorten that down to just the Kelvin cycle, with apologies to Dr. Benson. But basically, we see how NADPH and ATP are getting fed into that reaction, that cycle. And It's the, now we're talking about the CO2 coming in and getting acted on in order to make carbohydrate. So we're still zooming into the chloroplast. Now we're in that watery portion that we refer to as the stroma. So that's the overall big picture reaction that we've already talked about. So this is all called the Calvin cycle. It's called carbon fixation. It's called the C3 cycle. It's all the same. Sometimes it's referred to as down low there. It says photosynthetic carbon reduction cycle. But if you put that down into an acronym, it's PCR. What do you think about when I say PCR? Yeah, and so that one kind of trumps this. And so I don't use that acronym at all because I can't not think about the polymerase chain reaction when I say PCR. So we're going to be, you'll hear me say Calvin cycle or C3 cycle. That's usually how I refer to it. Okay. But just be aware, some people might say PCR and then get you all confused, in my opinion. So, okay, why do they call it the C3 cycle? Because when you look at the kickoff to the cycle, the first stable molecule is 3 carbons long. So that's why it was named that. Now, this is something that you're going to see. There's key steps. You got to use ATP at certain steps and NADPH at certain steps. And so then here's the big question. What does CO2 react with? So this was a conundrum that confused scientists for a long time because they could track that it was a 3-carbon sugar that was the first stable product. So they said, okay, well, carbon dioxide has 1 carbon, so it must be a 2-carbon molecule that it reacts with, so you can get to 3 carbons. And so they were hunting and hunting and hunting for a 2-carbon molecule that would react with CO2. Couldn't find it, couldn't find it, couldn't find it. And then finally, Dr. Melvin Kelvin, that's his real name, Dr. Melvin Kelvin came along and he said, well, there's more than one way to get to 3. What if you go up to 6 and then divide by 2? That gets you to 3. And so they went, oh, all right, let's look for a 5-carbon. And lo and behold, they found one. And so the 5-carbon is called ribulose 1,5 bisphosphate. So that means it has 2 phosphates at the 1 and 5 carbon. You can abbreviate that down to RUBP. So this is a sugar. It's 5 carbons, 2 phosphates. So the problem they had when they were first doing this is that they didn't know there was a 6-carbon molecule first formed. It was so ephemeral and unstable that when RUBP and CO2 react, it makes that 6-carbon, and then that 6-carbon quickly breaks down into 2 identical molecules of this 3-carbon sugar. And so that happened so fast, and technology back then, they didn't notice that 6-carbon. So because of Dr. Kelvin's contribution, he won the Nobel Prize for the Calvin cycle. That's why it was named in his honor. And so it was a group of scientists, but it was his contribution that really broke open the completion of figuring this all out. And so this is what I call the pie chart figure, the pie chart figure of the C3 cycle. So I'd like you to know these big terms that are being shown here. So the first step is the carboxylation step. So that's where you see the kickoff. You have CO2 coming into the plant through the stomata, and it's reacting with RUBP. Okay, so that you can, by the way, you can just abbreviate down. You don't have to write it all out. Okay, RUBP. And then the 3-carbon is right there. That's your stable product. 3-phosphoglycerate. Another way to say phosphoglycerate is phosphoglyceric acid. Anytime ATE is on the end of a word, it's an acid. So phosphoglyceric acid gets abbreviated down to PGA. So you can just abbreviate it to PGA. Okay, when you look at the carboxylation phase, do you see any ATP or NADPH? No. So what that's saying is, if there's enough CO2 and RUBP around the enzyme that pushes this reaction, it's just going to happen spontaneous, which is good. That's a good thing. So PGA gets formed and it doesn't need any input of ATP and NADPH for that. But then you can see the next phase, which is called reduction of the Calvin cycle. All right, reduction. Now you start to see ATP and NADPH needed. So in order to go from PGA to G3P, so glyceraldehyde 3-phosphate, we abbreviate to G3P. In order to do that, you need the input of ATP and NADPH. So that's the reduction. So the reduction is the idea that it's using NADPH to reduce, which means NADPH is giving off electrons to help this reaction go to completion. And you have ATP donating phosphate. Okay, so then the last stage is the regeneration. So when they are talking about regeneration, what they mean is you have to make more RUBP to keep this cycle going over and over and over all day long. And so you see this glyceraldehyde 3-phosphate, and then you see this big swooping arrow to RUBP. There's a lot of stuff happening in that big swooping arrow section, which I'll show you. But again, you need ATP. So ATP is needed both in the reduction and regeneration phases, and then NADPH is needed in the reduction phase. But if everything goes okay, you'll get this cycle happening over and over and over all day long. But you need to make sure that there's enough RUBP sitting there waiting as the CO2 comes into the plant. Okay, so this is what I call the pie chart of the Calvin cycle. Another thing to see here is that this glyceraldehyde 3-phosphate that gets formed, you're kind of seeing that as a branch point of the path. You can go in two different directions. So you see that some glyceraldehyde 3-phosphate goes to make more RUBP. And then they're also pointing these arrows down. So they're saying it can also, G3P can also go into a separate reaction pathway to either make sucrose or starch. So you need a fair dose of G3P made for all day long, because some of that has to go toward RUBP, some of it has to go for starch or sucrose. And the plant has to constantly assess, where should it go? How do I decide, how much should go? And so there's a lot of control on this that will coordinate this really nicely, so that all portions of this are going along okay for the plant. Okay, so the carboxylation reaction is being shown with that top bullet point. And the enzyme that allows for this to happen is called Rubisco. Its full name is called ribulose 1,5-bisphosphate carboxylase oxygenase, which is a mouthful, so I'm so happy they shortened it down to Rubisco. Okay, so anytime you see ASE in a word in science, you know they are referring to an enzyme. So we have carboxylase oxygenase. So this is a funky enzyme because it can, carboxylase means that it can react with CO2. Oxygenase means it can also react with oxygen. So it's got a dual function. So please try to get straight right away in your head what Rubisco means and what RUBP means. I've seen students confuse those, and it's really, really sad on the exam. So please don't do that. The way I remember is that CO, if I can remember the CO portion of Rubisco, that stands for carboxylase oxygenase. And then I go, oh, okay, I know that ASE means it's an enzyme. So Rubisco is the enzyme. RUBP is a 5-carbon sugar. Very, very, very different. So get that straight, please, because you don't want to confuse those on a quiz or anything. Okay. By the way, it's the most abundant protein in the world. When you think of all the organisms that do photosynthesis, they all have Rubisco. So it's not just terrestrial plants, you have algae, cyanobacteria, so it's a big deal. Lots and lots of rubisco on this planet. You look at terrestrial plants and their leaves, it's about 40% of the protein in the leaf tissue. So super abundant, super important, and yet it's kind of weird because we say it's sloppy because it can take in CO2 into its active site, but it's okay with oxygen too. It's like, yeah, whatever, I'll take either one. So the carboxylation reaction is being shown here in more detail because this helps you learn this process. So you see the CO2 coming in and reacting with the RUBP sugar. And you can see it attaching. And then you get two identical molecules of PGA. So it's the 6-carbon one in the middle. That's the one they didn't pick up on initially. They didn't see that in their experiment. All they saw was the PGA. Okay, so you can see in that reaction pathway, there's nothing to do with ATP and NADPH in there. It's not until you get to the reduction phase that you see that. And so PGA will go to G3P. The full reaction pathway is listed in that second bullet. You don't need the full reaction, you need to know it. Just know that PGA goes to G3P. However, you do need to know that you have to use ATP and NADPH in this reduction phase. And so often this G3P, they'll call it triose phosphates. That's, when you look down low here on this diagram, it says triose phosphates. That's kind of a generic way to say, in this case, G3P falls into that category. So try not to get confused on that word. Okay, so what they're showing in this diagram is that you do get some G3P exported out to the cytoplasm. That is where you're going to get what happens is G3P gets converted to DHAP, dihydroxyacetone phosphate, and then it goes into the cytoplasm. Okay, so then it goes into the cytoplasm, and then it makes sucrose. That's what some of the G3P is used for. So sucrose is made in the cytoplasm. However, when they actually track what's going on, most of the G3P is reserved fat. It stays in the chloroplast and is reserved for making more RUBP during the day, especially because that CO2 keeps coming in, and it needs RUBP to react with. Otherwise, this would quickly become very inefficient. Okay, so with the reduction phase, this is what we have happening. We have two of those PGAs, so they're only showing one just so it won't get confusing. So here's the PGA, and you see it gets phosphorylated by the ATP, and then NADPH donates off an electron, and you're left with G3P. Okay, so then once you have that G3P, most of it will get used to regenerate the RUBP. And so, by the way, sometimes they refer to RUBP as the acceptor molecule. What they mean there is that it's going to accept CO2 and react. So you see that in a lot of readings. They'll talk about the acceptor molecule. I don't know why they can't just say RUBP, but anyways. So that last swooping line on this pie chart is actually, there's a lot of reactions that are going on there that they're not showing. And when you look at it, there's 4, 5, 6, and 7 carbon sugars involved in this whole process. And when you look at, okay, what's going on in regeneration, the idea here is that if you have a pool of 6 G3P molecules, 5 of those will get used to make 3 of the RUBPs. And so I'll show you what I'm talking about here in a second. So this idea of the regeneration is, okay, we got to make more ribulose 1,5-bisphosphate. That's 5 carbons, but G3P, it's 3 carbons. So somehow you have to combine a bunch of the G3Ps and make 5 carbon RUBP out of that. And so there's several reactions that are going on in order to get the RUBP. So I wanted to also point out with this one, it's similar to the pie chart I've been showing you, but they're a little bit more detailed here where they show that sucrose is happening in the cytoplasm. Starch synthesis stays in the chloroplast. So you have 2 different physical locations for the long-term storage of carbohydrates as starch versus sucrose that will usually immediately get acted on and translocated through the phloem. Okay, so regeneration. This is the entirety of the C3 cycle, but that pink portion, that's the last phase. That's the regeneration. So you can see there's a lot going on here. And you can see that there's 4, 5, 6, and 7 carbons in this process. So that's the C3 cycle. They've color-coded it for the different phases of the pie chart. So the blue, the carboxylation up top. And then you have the carboxylation is the blue part, the greenish part is reduction, and then the pinkish part is regeneration. So it matches the pie chart here, right? So that's kind of nice. It helps with that. This is another version of that from a different edition. I put both in for studying purposes. It's the same thing, but maybe one clicks better with your brain than another. The one that clicks with my brain is this one. It's a little bit simpler. So this is usually the one I teach off of, but the other 2 are certainly fine to study off. So this is, again, the entirety of the C3 cycle. So if you look at it carefully, in order to get a pool of 6 G3Ps, in order to do that, you end up working with thinking about it in the idea of, well, let's take 3 CO2 and react them with 3 RUBP. And then if you do that, you'll end up with 6 G3Ps. So they're just doing that to get it to be even numbers, and it works out okay for showing what's happening here. And so this first step here, this is the carboxylation that's happening. They're not showing the enzyme, but that's Rubisco sitting there allowing for RUBP and CO2 to react. And then you get that PGA. And then this next step here, going from PGA to G3P, that's your reduction phase, where you use ATP and NADPH. And then you end up with 6 G3Ps. Okay, so now all of the remaining is the regeneration. So they're saying, let's take one of those G3Ps. One of them is going to be used for export to make sugars, sucrose. But okay, so let's ignore that last G3P. All the rest are going to be used for RUBP. So what are they showing? Well, if you combine, remember now, G3P is 3 carbons. So if you combine one G3P and another G3P with each other, that's 6 carbons. So that's what they're showing here. That's a 6 carbon. And then what they're showing is that you can have that 6 carbon react with another G3P. Okay, so that's 6 carbon and 3 carbon. So now we're up to 9 carbons. They're not showing that 9 carbon on this picture because it immediately splits into a 4 carbon and a 5 carbon. Okay, so the goal here is to get a 5 carbon RUBP. Well, you see one of these products is 5 carbons, so we're getting close. Okay, that one we're getting there. But let's hold off on that one. So now this erythrose 4-phosphate here, it's 4 carbons. That's not good enough. We're trying to get to 5. That's the goal. So if you take that 4 carbon and combine it with another G3P, 4 plus 3 is 7. So this is a 7 carbon. This 1 7, so you have heptulose 1,7-bisphosphate. We're not quite there, but if you combine that with another G3P, now you're up to 10. Split that into 2, and you end up with a ribose 5 and a Ziose 5 phosphate. So now you're getting close. Like, okay, now we have these 5 carbons, but now we need to create RUBP from those. So that's where you see these arrows converging now. What you see here is those 3 molecules there will get converted to ribulose 5 phosphate. So now we're getting even closer. It's really close, but that's not the same thing as RUBP. You need one more phosphate, one more. So that's where the ATP is coming into play. If you phosphorylate ribulose 5 phosphate, then you're up to ribulose 1,5 bisphosphate. Now you've completed it. So for learning this C3 cycle, knowing the carboxylation phase, yes, I want you to know that really well. Knowing the reduction phase, yes, I want you to know the details of that. This last bit here, the regeneration, I want you to know what I call the numbers game. The numbers game. I'll post that into, I don't think I posted that yet, into the EduCat, but let me pull that up. So I want you to think about the regeneration when you look at it really quick here, and I'll go over it next time. When you look at it, basically it's a numbers game, and I'll work through this next time. But this is kind of, whoops, sorry. Hang on, let me just show you this real quick. I'm going to show you how to get to this idea. This is more like what I want you to know. You don't need to know all the different molecules or the enzymes that facilitate those reactions. You need to think about, how do I get to 5 carbons? And then how do I convert those 5 carbons into RUBP? Okay, so that's what we'll go over this next on Wednesday. So on Wednesday, come prepared to ask questions after we go through more of the C3 cycle.